Breast cancer is a heterogeneous disease that will affect 1 in 8 women during their lifetime in the United States.
Resveratrol is a natural compound that originates from red wine. It has a similar structure to estrogen, which makes it more likely to alter the receptor activity and inhibit cell growth. This has also been shown to have anti-cancer properties. Due to its low bioavailability, it is not a great therapeutic option, so our research group has synthesized derivatives of resveratrol with the goal of increasing the anti-cancer activity and bioavailability of the compounds.
Previously, our lab screened 28 resveratrol analogues. From that work, we found that the compounds that significantly inhibited cancer growth in our breast cancer cell lines contained methoxy groups at specific locations on the analog. These were at the R3 and R5 position. We also noticed that the cis conformation of these analogs seemed to be more active than their trans counterparts.
We have screened an additional 52 analogs that were synthesized based on the structures we tested before by altering the functional groups to determine the activity in the cell lines. 16 were found to significantly inhibit cell growth and proliferation in both of the cell lines. All the analogs can be seen in the graphs, ranked from most effective to least effective.
The following patterns demonstrated to have the most anti-cancer effect on ER-negative cells. Compounds with methoxy at R3, R5, compounds with trimethoxy at R3, R4, R5, and compounds with bromine at R1 or R4.
All of the experiments were compared to a controlled treatment that was normalized to 100% for cell viability. All of the bars that are below 100% inhibited cell viability. Over here are the methods that were used to conduct the experiments.
When we analyzed the data, we found that the activity of the analogs is dependent on the specific functional group at a specific location. Specifically, methoxy groups at the R3 and R5 on ring 2, and the bromine functional group at the R1 on ring 1 and R4 on ring 2 seem to be the key to the analogs activity. We don't have any data on the specific mechanism behind the activity of these functional groups, but overall, these functional groups may increase the uptake and bioavailability of these compounds.
We are currently testing the analogs that were significantly inhibiting the cancer cells' viability in dose response experiment to determine the lowest effective dose. More analogs are also being synthesized so we can continue to study the effect and mechanism of the specific functional groups on the analogs activity.
Thank you, Florida Undergraduate Research Conference, and thank you, Dr. Rhodes, for being our faculty mentor.